Hello and welcome to SciLux, the podcast where we talk about scientific developments and technological changes in Luxembourg. Not always, but today in Luxembourg, which means we are proudly powered by Research Luxembourg and recording at the Media Center of the University of Luxembourg. And our guest today is Brice Appenzeller, who is the head of Human Biomonitoring Research Unit at Luxembourg Institute of Health and Associate Professor at the University of Luxembourg. Brice's research focuses on biomonitoring human exposure to pollutants. Brice, thank you so much for coming today.
Thank you, Anna. I'm happy to be there.
Okay, so we're going back to exposure. I have to say that we already discussed it a little bit, exposure to pollutants, to unknown chemicals and everything like that. If you're interested, we had Dagny Ulrich on the podcast as well. We were talking about exposomics, but I still wanted to go back to this because I don't think that everybody listened to it and everybody remembers what it is. So what is exposome actually?
Exposome has actually been defined by Christopher Wilde. It's already more than 10 years ago and is considered to be the sum of all the physical, microbiological and chemical agents to which humans might be exposed over the life from conception to death. That's the official definition.
No pressure, right? That's probably a lot.
It is.
Exactly. And also I like the fact, as a real scientist, you said might be exposed, right? Not is exposed.
You noticed that.
Of course I do notice that, It's always like I love when I get the answer, it depends. It's more or less the might is also important here. Okay, so what are they then? What are the environmental agents or factors that we are exposed to?
If you think about microbiological agents, you could, of course, think about bacteria, viruses. If you think about physical agents, you could think about radiation, which is a typical physical agent present everywhere. And our specialty actually concerns chemical agents and more specifically chemical pollutants. So that can be a lot of things from all the things that are present in the room. For instance, you could think about there is some carpet, there are some flame retardants, there are some plastic compounds in all the devices around us. In your food, you could think about all the things that migrate from the packaging to the food. You could think about all the pesticides that are used to produce food, vegetables, fruit, of course. You could cross the street and be exposed to diesel exhaust, and all these things compose what we call the chemical exposome, which only concerns the chemical agents.
What always fascinates me is when you are a scientist specializing in a field like that, it's one of those fields when we tend to forget, right? We all know, we read about it, yes, all these exposures, but then we go on with our lives. But if you do it on a daily basis, it must be difficult to function, because you always realize, or you don't, or you're also able to kind of shield it off, or you think about it all the time.
I totally agree because that's invisible, so you might start to forget about that. That's probably one of the most problem with this job is that you always tend to think about all these exposure around us. And I have to admit that I became a bit paranoid with years because of that. Yeah.
Also, when you go to a shop, right, and so there's still so much plastic, all the containers we are buying, right, probably reading all what it is made of and what it means is a bit more stressful for you.
It is. And on top of that, I think I am communicating this stress to people around me very easily.
So the family is also involved.
Absolutely,
then, of course, as usual, it's like my family.
Me for that.
Of course, do you know, the kind of typical situation at a Christmas party, right? When you're not supposed to talk controversial subjects, but it always kind of slips in. So we know that's the exposure, but then we have to figure it out, find it out, right? So we say we are exposed to this or that, but then how do we see what we were really exposed to? How do we analyze it? How do we test the exposure?
Actually, that's the problem because you don't see it. So to show exposure, there are indirect approaches that are still based on analysis conducted on air samples, water, food, soil, and by means of extrapolation, you could roughly assess what could be the transfer to human. And there are the direct approaches, which are what we do actually, which are called biomonitoring, which consist in the analysis of these chemicals or their byproduct, I mean the product transformed by the body, into biological samples directly collected from people. So most of the time, it has historically been urine and blood, but we also developed the possibility to analyze that in hair samples, which is also a very interesting type of sample to analyze all these chemicals.
Absolutely. So if we look at those 3 different types of samples, does it mean you're testing for different types of chemicals, or can you find the exposure in all 3 of them?
Both, actually. Sometimes some chemicals could be detected in urine, but also in blood and also in hair. But there are, of course, some specificities, and we have some studies currently highlighting that actually what we call the ghost compounds that are not detected, let's say, in urine samples, but that would be detected in hair samples collected from the same individual, which means that if you only analyze one type of sample, you will miss a part of the information. And that's why we are working more and more with a view to develop multi-matrices approaches to analyze these chemicals.
Which eventually means that you kind of can confirm the presence of certain chemicals with 2 different types of samples. And sometimes, as you said, the ghost ones, you just have them only in hair.
Absolutely, yeah. So you can confirm, and of course it's very interesting, if you can find the same chemicals in 2 different types of samples from the same individuals, but the absence does not always mean the absence of exposure, because maybe just because of the specific physicochemical properties of these compounds, it is not incorporated in this matrix, but you will find it in another one.
So we can't really talk about the best method.
Not really. I would like to have the ideal matrix, but the experience shows that the more samples you have, the more comprehensive, the more complete will be the information.
And on top of that, of course, all the chemicals, they interact with each other, so you can have multiple exposure. That actually results in something more than if you just had one. And I think this is the good moment to ask the pub quiz question that you prepared about your research exactly on that.
Absolutely, yeah. Actually, we know now that single exposure, unfortunately, does not exist, and that everybody is exposed to many different chemical pollutants simultaneously. And that's my question for today. For you, do you have an idea of what is the number of chemical compounds to which humans might be exposed, let's say, on the daily, simultaneously? This is something we tried to assess recently in a recent article. I can give you the information later.
Later, exactly. So remember, yes, please don't spoil right now. We will try not to discuss it before we reveal it. So the answer only at the end of the podcast. And it's also interesting because, as you were telling me, it's on real data, right? And once again, it's just one analysis article. It means that it will be reconfirmed and checked whatever. We're just talking about that recent publication of yours.
Yeah, that's actually an approach that we developed because you can turn it differently. You could consider all the different chemicals that have been synthesized and that are registered in chemical agencies. But of course, everybody is not exposed to all the chemicals, and some of them are produced in very low, limited amount and are not really relevant. But that remained indirect approach. And of course, the idea was to start from analytical results, so the results obtained from the analysis of biological samples collected from people, and to extrapolate to what is this real number.
So once again, answer at the end. And of course, we will also share the link to the article, I guess. So if people want to read more, they can check it out. Okay, so one of the things I remember I was discussing not only with Dr. Dagny Orich, but also with Professor Emma Szymański about unknown chemicals, is the thing that, I always thought, all right, you have a sample and then you see what are the chemicals. And it's actually the reverse thing. You test for chemicals. You need to know what you're testing for. Let's put it this way, right? Am I right?
You are, absolutely. There are different approaches, what we call the non-targeted approaches and the targeted approaches. And of course, Emma is more specialized in non-targeted approach, which is very interesting as well because it can help to discover new relevant compounds. But our team is specialized in targeted approaches, which means that we develop what we call multi-residue methods in which we specifically target all the compounds. So we include more and more. So I like to say that the methods are living methods, because if you would have seen our methodologies, let's say 15 years ago, we were targeting, let's say, 10 to 20 different chemicals. Now, with the methods that we have, we target more than 160 different chemicals in one single compound. Of course, it means that we will only detect what has been included in the methods. Of course, some of the compounds that are in these methods will not be detected. But what is interesting is that we know what we are analyzing. And in the case we don't detect a chemical, we can say, okay, we were analyzing this one. It's not present in this type of sample.
So how do you choose those chemicals, those 160?
We based on priority list. We based on what is commercially available, because you need to have standards that you can buy to develop your methods. There is a part of serendipity. Sometimes, okay, we have this standard that is in the mix with other ones that were relevant to us. So, of course, we will keep them. And that can happen as well. Sometimes you have a good or bad surprise, depending on scientifically interesting, but that's not really a good thing regarding public health. But most of the time, we know that, okay, if there are some specific problems or attention, if the focus is on a specific chemical family for its toxicity, let's say neurotoxicity or associated with developmental issue, so we will include these compounds in the methodologies.
And we are also talking here about the endocrine disruptors, right? That you sometimes mention, or not?
Yes, but actually endocrine disruptors can be all and everything.
Okay,
and
great.
People usually hear things like plasticizers, like bisphenol A, which is a typical known endocrine disruptors of phthalates. But you have to know that much more chemicals have endocrine disruptor properties. Just they are not famous, because there is not enough studies on these compounds, but they can be endocrine disruptors. And we also have to keep in mind that endocrine disruption is not the only toxicity mechanism. And many other chemicals are not endocrine disruptors, but are toxic as well.
Of course. And also that's a question of this holy grail of understanding, okay, these are the chemicals, this is what we think they cause or they might cause, but we're not really sure. Is there anything we can say about diseases and our exposure? That's really, this is, okay, one-to-one related, might be related, let's put it this way.
It's true, I totally agree. That's very complicated, because to show the job that is done by risk assessment agencies to demonstrate the toxicity and the association between specific disease and specific exposure usually requires several steps. There is studies on simplified models. I mean, animal experimentation can be part of the job, of course, or tests on cell cultures as well. But usually it's also necessary to have some kind of confirmation on the human. And of course, you cannot voluntarily expose human to chemicals at controlled levels. And it's epidemiological studies in which you have to assess retrospectively the exposure that is responsible for the adverse effects that you observe today. So that's very complicated, and it can take 10, 20, 30 years.
Okay, so we can't really, I understand that was a long answer to say I can't give you a direct answer.
Unfortunately not.
Okay, good. No, just checking in case I didn't understand something. Okay, so we have both the exposure to different chemicals. We will discuss later on the fact that they kind of intertwine and influence each other and whatever, and we can have multitude of exposures. But there's also one more thing, the target response. So this idea that maybe someone does not react to the exposure as much as someone else, right?
Yeah, absolutely. That will depend, and that also complicates even more the answer to your previous questions, because, of course, there are some people who will be exposed to some chemicals and will never be sick because of that. We all know smokers who are absolutely healthy. But we know that, tobacco smoke is a well-known carcinogenic compound. And that's the same for all the chemicals. And on top of that, the levels of exposure are usually lower. But of course, the consequences will depend on your genetic, but on your specificities. I mean, your specific weakness, your metabolism. And we usually consider that the weakest population are the young children and the fetus, of course. And that's why assessing exposure of pregnant women is very important as well. But we know that it really depends on individual specificities, because on top of that, sometimes the same exposure will lead to different disease, because you have this specific weakness and other people might have other weakness. So that even more complicates the situation.
So how do you get actually same exposure? Is it, analysis of twins? Or how can you make sure that the exposure was the same?
You can never be sure.
Okay.
That the only thing that you can do is collect samples and assess the exposure.
Okay. All right, I understand. So you mentioned the younger populations. I think it's a moment to move to one of the studies that you made. And it's one of the studies, I really read the Luxembourgish media, and it's very rare that you have really a lot of science in there. So it's one of the studies that really went into media as well, was discussed. So when you look at the hair samples of youngsters, children, I guess, right? Children in Luxembourg. So what was the study about and what did you find out in Luxembourg?
Yeah, that was a study that we conducted on Luxembourgish children, a bit more than 250. It was conducted during the COVID period. So
lovely.
Collecting hair samples was very convenient because that could be collected by the parents and sent to the lab directly, avoiding the contact. We analyzed the hair samples that we collected from these children. The method that we used was targeting at that time 150-something chemicals. And if I remember well, the average number was 88 compounds per children. So that clearly shows the multiple exposure. And I think that the lowest number of chemicals was close to 30 per compound for the luckiest kids.
That's not great, I would say.
That's reality.
Yeah, well, exactly. It's just kind of confirming what we could think, I guess. But one of the things I noticed there in that study, and well, you mentioned already BPA and you said this is not the worst, but still, it's one of those things that does get to the general public, and we are proud to buy BPA-free products and say, okay, then the exposure is gone. But basically that was, from the perspective of your data, it was the highest median concentration was of BPA.
It was, yeah.
So why? Where does it come from?
It's present everywhere. Actually, it's present in most of the plastic materials. It's a direct compound of polycarbonate, epoxy resins, and it has been bound in baby bottles only. It was still allowed in many other compounds at that time, so that explains the exposure of the population.
So
it's still present.
We just limited it because, exactly as you're saying, baby bottles, toys, I guess, have those labels, but that's it. It's everywhere else.
It is. And even when you ban a compound in some food device, it will take years before it really disappears from your environment.
Not mentioning the fact that, as you said, that's really one compound that we are aware of, right? But it doesn't mean that the industry will not decide to make something a bit different, that it's not this anymore, right?
It's exactly what is happening.
Exactly.
Because by the way, it's very interesting because we are analyzing different compounds because bisphenol, it's a wide family. There are bisphenol A, of course, but bisphenol B, bisphenol S, bisphenol F. And it's interesting to see that in parallel to the slight decrease in bisphenol A concentration, we already observed a couple of years ago the increase in the concentration of bisphenol B, which is what is used by the industry to replace bisphenol A. So it's already there.
I have a feeling we're not very positive today in the episode, but still, we're talking about hair samples. So I just wanted to ask you, because one of the research, that I did was very interesting for me, because I found out a new society that I have to say I was not aware of. So the Society of Hair Testing, and you happen to be the vice president of it. And I really got into one of my favorite rabbit holes, reading on how you're supposed to prepare hair samples and all that. So there was, let's say recently, more than a year ago, some recommendations published by the Society on hair sampling. So how do you prepare it? Because again, when I go test myself, normally it's urine sample or blood sample. I'm kind of aware already how does it go. But with the hair samples, what were you telling the parents, for example, during COVID times? How were they supposed to get the hair from the children? What do you do?
Yeah, actually, we prepared a tutorial to explain them, okay, you will collect the hair strand on the back of the hair, close to the skin with scissor. You will wrap it in aluminum foil and send it in an envelope. So that was clearly explained with pictures, actually, to make it even easier for the parents.
So what is crucial then when you're getting hair samples? I understand back of the head.
The back of the head, what we call the vertex posterior. That's actually also very important because you have to know that we use hair analysis to analyze chemical pollutants, but it's used a lot and for longer time in forensic toxicology to analyze illicit drugs, biomarkers of alcohol consumption, like, let's say, or for the drug cannabis, cocaine, and so on. And then it's very important to collect in very specific manner. So in this consensus was described all the steps that have to be followed: collect at the back of the head, submit to decontamination to remove what has been possibly deposited on the surface of the hair.
So we're talking shampoo, basically, for example.
Yeah, somehow, it looks like that. Mm-hmm. And to make sure that the results can be interpreted correctly.
But they can be cut with scissors. It's not that you're actually pulling hair from people. I don't know. Sometimes sample collection might be also difficult. So that's
we don't need that.
Okay, that's good. You don't need the full hair, let's say, for that. And how much do you need? Is one enough?
There are some studies trying to use only one hair, but that's very rare, and that's not the use in routine analysis. Usually you need a strand of hair, let's say, of the size of the thickness of a pen.
And by the way, you just reminded me that if listeners are interested more in biosampling and in general what is happening in Luxembourg, I also did a very interesting interview with Kathleen Momaerts from the biobank and how the samples are prepared and that they are actually sent by some couriers that I was not aware of. So listen to that one too. And as you mentioned, forensic medicine, I also had an expert from the Luxembourgish police. We were talking about fingerprints, but still very interesting how the samples are prepared and what you can know from fingerprints. We will share both of them, of course, in the show notes. And now coming back, so we have the hair. You said aluminium foil? Why aluminium foil? What's the best way to store?
Just because aluminium foil will block the hair, so the different fibers will not move and stay aligned. And then if you need to do segmentation, to do a history of exposure or consumption, it's the best way to block the strand. And on top of that, it will be protected from the light. So the compounds present in the hair strand will be really stable for months or even years.
Also, when I was in that rabbit hole, I was pretty surprised how human-centered we are. And I am trying not to be, but still I get into that trap. And I just never thought, okay, we're getting hair from humans, of course. That's what we're interested in. And then I discovered that you do this with animals as well.
We do. Because actually it's the same route of incorporation. So from the blood, actually the compounds are incorporated at the level of the hair bulb, which are living cells, contrary to the fiber, which is composed of dead cells. So the chemicals are transferred from blood to the living cells, and it's trapped in the hair cells when they die. So it's the same process in human and in animals, of course. So we did that in several studies, actually. That was in one collaboration, if that's the one that you saw with chimps in Uganda, where we demonstrated that actually the chimps who feed on all the crops around their territory. And of course, the crops are treated with pesticides, and eventually the chimps are also exposed to pesticides. So we helped colleagues from the Muséum d'Histoire Naturelle de Paris who did this study. And we also collaborate with colleagues working on the rodents, the mice in the crops. And we could show that the protection areas where pesticides are not supposed to be applied were often not sufficient to protect the microfauna. And despite of these protection lines, the mice were still exposed because all these pesticides were still present. And we could also help to demonstrate the transfer from insects or worms to these microrodents. So the contamination of the food chain because of the treatment.
Very interesting. And then where do you get the hair from? I mean, which sample, where do you, because it's not back of the head, I guess, or it's also important?
The back of the mice, yeah. And for the chimps, it was much more difficult because it's not allowed. So the colleagues has to climb in the trees to go to the nest of the chimps and collect the hair that were left in the nest.
Okay. While watching whether they're not coming back at that moment, I guess as well, somebody else was there on the lookout. Okay. And once you get the hair in the laboratory, what happens? I mean, how do you actually check what is inside?
So there are different steps, the pre-analytical steps and the analytical steps. So starting from a strand of hair, there is, as I mentioned, the decontamination to get rid of what could have been deposited on the surface, which is not representative of the internal dose. It has not been incorporated from blood, so it's not relevant. So we wash the hair to remove that, if any. Then usually hair will be pulverized to increase the contact with the solvent that we will add and the particles, the hair. Then, depending on the chemicals that will be analyzed, we will use different type of solvents to correspond to the physicochemical properties to extract these chemicals from this hair powder to this solvent. And then there is different steps of purification, concentration, and so on. And then the extract will be analyzed on different types of machines based on liquid or gas chromatography with mass spectrometry for the detection.
Of course, mass spectrometry is there always at certain
It's
moment.
always
Absolutely
there,
important, yes. Also in space research, which is also interesting. I mean, different type of spectrometry, but we are there too. Important method, always fascinated by it as well. Okay, I think we discussed quite a lot of different things on the hair sampling and how you work and everything. And the last remaining thing that I got interested is the new doctoral training, is it? Doctoral training, yes, that has just been started, right? It's like end of last year, more or less September, something like that, if I'm not mistaken. Yes. And it's called Expose, right? Of course, we're talking about exposomics and exposure to different pollutants and other things. And I was just wondering, as you're doing it also with my former guests, so with Dr. Guy Fagherazzi, what are your plans? What kind of PhD students are you trying to prepare there?
So the Expose DTU, Doctoral Training Unit program, is really focused on the exposome because, as we discussed in the beginning, exposome is composed of many different things. So the idea is to investigate different things. On the one hand, the different physical, microbiological, and chemical agents to which you might be exposed. So that will be a part of the research project of some of the PhD students. On another way, we will investigate what we call the determinant of exposure, the socio-demographic parameters, of course, depending on your job, depending on your habits, depending on the food you like, on your physical activity, you might be more exposed to some agents and not to other ones. So the determinant, it's very important to understand the determinant of exposure, to identify what are the sources of exposure. And finally, another pillar will be focused on the assessment of the biological effect associated with exposure. So that will be the three pillars of this program. And the idea is to provide these students with what we call a hybrid expertise. It means that that's a bit in opposition with previous approaches, because for this complex thematic, most of the time you need to have different skills, and that's really what we want the students to obtain. They will be specialists in different fields. For instance, be able to analyze the data, let's say work on the advanced statistical methodologies, artificial intelligence, but also work in the lab to know how the samples are analyzed and how they are produced. So that's really the idea of this program, to teach the students, to train people with this multiple expertise that will help them to better understand the field and to communicate with the different partners.
That sounds very interesting. So if you're into that, I guess you have to investigate more yourself if you're planning to do a PhD maybe, or you're just starting on a master's level, you know where to go next, right? Let's put it like that. And what remains is going back to the pub quiz question that you asked in more or less beginning of the podcast. It was about the exposure, right, to different pollutants at the same time and how many they are, right? I remember well?
Yeah, exactly. as I mentioned, when we analyzed the hair samples from the Luxembourgish children, we detected close to 80 different compounds. So we use these results, but many other results, mainly based on hair analysis, by the way, from population in Luxembourg, in France, in China, in different countries. And based on our extrapolation, we consider that the number of different chemicals to which humans are exposed simultaneously might be at the minimum more than 200 and possibly up to 3,000.
That doesn't sound good. We're not gonna finish on a positive note. But that also means that they must be, influencing each other.
Absolutely, yeah. And that also complicates the understanding of the adverse effect associated with the exposure. Because everybody now heard about the cocktail effect, and we know now there are very, very clear evidence in animal experimentation, cell cultures, as I mentioned, that demonstrate that the effect of a combination of different chemicals can be completely different from the effect, the toxicity of each compound taken separately. And that's also very important because for the risk assessment agencies or European organisms like EFSA, European Food Safety Agencies, all what we call the limits, like the daily acceptable concentration intake, are based on toxicity studies conducted on one single compound. And that changes everything because it means that since single exposure does not exist, it's always multiple exposure. It means that all this daily acceptable intake will have to be revised and definitely decreased. And we know now that for many chemicals, the level of exposure that was considered safe, let's say 20 or 30 years ago, are totally irrelevant today. And we know that some adverse effect can appear at much lower levels.
And how much do we know about them, working together? So how much they, amplify each other or...
That's a good question. Sometimes they could have the same target, hormonal receptor, if we are talking about endocrine disruptors, and sometimes completely different mechanisms of action. And of course, that's the principle of an army. If you attack on different side, of course, the effect will be higher.
Oh God, now we are in the army on top of everything with the positive. I'm thinking of the positive note. Maybe the positive note is just to be a very aware consumer as usual and take care of what you consume, where you go, what you buy, what you visit. Also here in the studio, you can't smell, but it's all the time smells of new things. So I'm always wondering what I'm exposed to when I'm here for that 1 hour. So it's better to stop exposure right now, stop the episode so we can go breathe a bit of fresh air, although this fresh air is also a bit questionable, I guess. It must be really difficult to be you. I'm thinking of that right now.
I try to stay optimistic as well, and that's exactly when I mentioned the study of the determinant of exposure, because I'm really convinced, because what we observed in the population, we observed some individuals with super high levels, super high concentration, but some other with super low concentration. And all of them live in the same country, somehow have similar lives. And it means that it's possible to have this low level of exposure, but we need to understand the sources and the determinant of exposure, and then we can be able to work on it to decrease exposure.
So I think what we're heading on to is the, as I say, boring recommendation, eat healthy and do sports. But because that doesn't sell very well. Everybody knows about it already. We would prefer a pill rather than anything like that. But still, this is very, very important. And what reminds us to thank you, Brice, very much for coming today.
You're welcome.
And this is it for today. Don't forget to subscribe, to follow us, to check what we are writing on all the social media, and check other episodes. We will share everything we discussed today in the show notes. This was Silax, and my name is Hanna Siemaszko.